Hi folks, and welcome to this shorter version of the producing new cells topic, which is part of Unit 2 in the National 5 Biology course. My name is Stuart White. I'm an experienced 20-year plus teacher of biology, and I'm going to be taking you through the topic just now and covering all the main points. So the first place to start off with when we're talking about cells, because cells are the building blocks of life, is to talk about the organisation within an organism and how those cells build up to become the multi-celled organisms that we are ourselves and that other plants and animals and so on are as well. So initially we have cells working together to form tissues, which work together to form organs, which work together to form systems, which then make up the entire organism. Okay, so we call that the hierarchy or the organisation of a multi-celled organism. We get cells which make tissues, which make organs, which make systems, which make the whole organism. And you do need to remember that hierarchy, and you do need to remember the order of each part as well within that. And the reason for that is because you can be asked about this in the exam where they give you a sequence question and they ask you to fill in the gaps, so make sure you know those really well. Now, cells they come in a wide variety of different specialisations, which means that they all have particular jobs or functions to do. And as a result, we need to have the ability to produce these many different specialised cells. Now, we've got a particular type of special cell within animals, which is called a stem cell. Okay, now a stem cell is an unspecialised cell. Okay, they are unspecialised, and these unspecialised stem cells can either divide to self-renew to make more stem cells, or they can become specialised, okay, and take up one of the many specialties that we have within a multi-celled organism. For example, we've got some red blood cells here, we've got some skin cells, and we've got some neurons as well. Okay, and each of them is very unique and different to the other ones, and we can only do that, we can only produce that wide variety because we have stem cells which are unspecialised and have the ability to become many different types of cells. Now there are two places that we find stem cells, and those are in the early embryo. Okay, so if we look at the cells of the early embryo, we'll find stem cells, or in some tissues within adults. So for example, in our liver, our bone marrow, we've even got cardiac stem cells in our heart as well. There are stem cells within adults too, and we call those tissue stem cells. Okay, so the two sources are embryo and tissue. And that leads us nicely on to the last part of this topic, which is all about how do we actually physically divide cells? How do we make new cells? Well, we do it through a cell division called mitosis. Okay, now mitosis is a division where we take one original cell and we produce two identical daughter cells. And we'll see in a moment when we look at the process how we get to that point. We start off with one parent cell here and we end up with two identical, that's an important point, daughter cells at the bottom here. So let's go through the purpose of mitosis. Why is it so important? Well, we need mitosis to maintain our diploid chromosome complement. Now, when we talk about that, we just mean to maintain the genetic material within the cell, to keep it the exact same as it is in the original cell and each of the new cells that are made. Okay, that genetic material needs to stay identical. So the phrase we use for that is we maintain the diploid chromosome complement. Now, the other reason that we need cell division, or mitosis specifically, is for growth and repair, particularly of like damaged or worn out cells that need to be replaced. Okay, so they need to be repaired, or we sometimes, go through growth periods in our life, we need cells for growth as well. Okay, stem cells are another example of cells which can help us with growth and repair as well. So mitosis involves 5 main stages that you need to know about for your exam, and we're going to go through each of those stages just now. It's in reality a lot more complex than this, but for you guys at National 5, you just need to know these 5 steps. So first of all, we have our thin thread-like chromosomes within our original parent cell. Now we want to make a new cell which is identical to this, right? all of the new cells that are made need to be identical to the original cell. Okay, we need to maintain the diploid chromosome complement. And we do that by going through 5 stages. The first stage, the chromosomes need to shorten and thicken. And we can see from this stage to this, the chromosomes have become much shorter and much thicker. And we can also see that they are now in this sort of X shape. Okay, now what's happened there is there's been some DNA replication essentially, but we've got 2 what are called chromatids, which are joined together at a central point like that. And those 2 chromatids, that's each of my arms, are joined together to form 1 chromosome. Okay, and that can be a little bit tricky to get your head around, but when 2 chromatids are joined together in this sort of X shape, it makes a chromosome. Okay, so we'll refer to them as chromosomes when they're like this, and we'll refer to each arm as a chromatid, and then we'll see what happens a little bit later on in this process. So after the chromosomes have shortened and thickened, they then line up at an imaginary line down the middle of the cell that's known as the equator. Okay, then the chromatids, so that's our 2 halves of our chromosome, are then pulled apart, okay, to opposite sides of the cell. Now they're pulled apart by these green thread-like structures here, which are called spindle fibres. You need to remember that term. Okay, so they line up at the equator, they're attached to these spindle fibres, and then the spindle fibres pull the chromatids apart to opposite sides of the cell. Once those chromatids are at the opposite sides of each cell, a nuclear membrane forms around each of them. Okay, so we've got a new nuclear membrane. That's the membrane that surrounds the nucleus within our cell. And then lastly, our cytoplasm has to divide, and it has to divide so that we're ending up with 2 new cells. Okay, so the cytoplasm divides or splits in half there, and what we're ending up with is 2 identical daughter cells. Okay, and if we look at those, they look very similar, in fact identical, to the original cell here, which has undergone mitosis. Okay folks, so I hope that's been useful. I hope that's helped you out in terms of understanding the producing new cells topic. I hope that you've learned a little bit today. If there's anything else you're still not sure about, leave me a comment below and I'll get back to you. Otherwise, well done, class dismissed.